Okay, so thank you very much, Geoff, for this introduction. So thanks also to the audience for your attendance. So this will be the first presentation on evidence synthesis. As you may have noticed, we have three main topics, and within this particular one on potential human and animal health effects, we do have four questions that are to be answered by the evidence maps being prepared in this work package. Okay, so this is actually the work of a team involving various partners, including, for instance, RIKILT from Wageningen, ABI from Sofia. We have partners here from Catalonia, from Krach and University of Girona, as well as JKI and, the Alpia Adria University. Okay. So, the schedule for this presentation is to start off with, how we developed, questions and protocols, to present you the main findings and observations that we actually made during the exercise. So these are preliminary results, because we haven't actually published the final outcomes, but we can also already show you some things in this advanced stage of development. And the various results that will also be highlighted in more detail are, the evidence map outcomes for these four topics. So we have whole foods feeds being tested in experimental animals. There the focus was on health effects, so not just performance, like body weight increase or feed intake, but also for instance, clinical biochemistry and such. Potentially changed endogenous allergenicity of GM crops. So some crops are known to be allergenic, like for instance soybean. I think other crops like peanut are also well known, but they haven't been genetically modified, at least not yet as commercial GM crops on the market. The question was, is that particular feature of the crop changed following genetic modification? Key compositional parameters, actually Armin already referred to that question, which was actually one that we adopted based on stakeholder feedback, for instance. So the compositional parameters of GM crops, it's one of the keystones, the key parts of the comparative safety assessment, the initial extensive analysis of the composition of a GM crop as compared to its near isogenic non-GM counterpart. Animal studies with newly expressed proteins. So this is very similar to toxicity testing with purified compounds, albeit that in this case we test proteins, which are of course biological substances, which can be degradable, for instance. But here the focus was on animal studies, so for instance particularly rodent studies, laboratory animal studies with those newly expressed proteins. And the background that we will explain further on, if there's some time left, the GMO safety assessment, because all this is actually done in the frame of scientific research, our project, but we actually have as one of our objectives to inform professionals, for instance, active in the area of GMO safety assessment, and of course also the wider audience of other stakeholders could of course draw information from the outcomes, as well as status quo and outlook. Okay, thank you. So this in a nutshell is how we developed actually the questions and the protocols. We initially started with 10 draft questions, based on what the various experts within the consortium thought might be interesting topics in the field of human and animal health impact of GM crops, like food and feed, but also through other routes of exposure. And this was actually submitted to stakeholders. We had a stakeholder consultation event in Berlin. Some of you may remember this because they are regular visitors of attendees of our stakeholder consultations. And based upon the feedback received, either on site or via written comments, we actually revised the questions, and the stakeholders were also allowed to prioritize the questions. So we got ratings based on different categories or topics that stakeholders, or features that stakeholders thought important. Our colleagues from GRAS actually ranked those questions, and we were able to pick the top 4 of those questions. And then the questions actually had to be incorporated into a protocol that actually described how we were going to actually collect the evidence, collect all the records that were relevant for answering the question. And also on the protocol, we had a round of stakeholder consultations. So eventually we ended up with a revised protocol. Okay. So the topics addressed, as already briefly mentioned, include, for instance, the effects of GM crop-derived whole food and feed on experimental animals. And I think it's important to stress that this is actually health beyond performance. So there are a lot of studies out there testing, for instance, GM crops in all kinds of domestic livestock animals, for milk production, meat production, and they usually test performance measures such as body weight increase, feed intake, carcass characteristics, for instance, if you talk of studies in broiler chickens, which is a very popular model, or pigs, but that was actually beyond our scope. We wanted to have studies that focused on other parameters besides those performance, including, for instance, hematology, clinical biochemistry, histopathology, and such. Okay. The other topic was changed endogenous allergenicity, as already explained. So much of the current methodology for allergenicity assessment focuses on the potential allergenicity of the newly expressed protein. I think EFSA also has this relatively elaborate description of how to test for endogenous allergenicity of host crops in its guidance. So it actually extends beyond what we call the Codex Alimentarius guidelines. So the question that arose is what is already known on potential changes in endogenous allergenicity. And the focus here was on allergic reactions, because there's a lot of literature already out there that might tell you something about the levels of certain allergens in a particular crop, and then part of that will be about those levels in GM crops. But we were wondering whether there was any study out there that would actually correlate those changes in the levels of endogenous allergenic proteins with reactions to that crop measured in allergic patients, patients allergic to the crop, or perhaps an animal model with animals sensitized to that crop. And why is that important? Because there was actually a kind of discussion going on at that moment, like EFSA requires this to be tested, at least changes in the allergen repertoire, but what does it actually mean in terms of risk assessment and risk management when it comes to, say, the risk of allergies in patients? Okay. The third topic is compositional analysis. And this question also evolved over time during our interactions with stakeholders. So initially we had a few questions, like on, omics of GM crops and the robustness of methods. Actually, we noticed that there was a preference for among stakeholders to focus on key parameters of crop compositional analysis. Because, you may have findings in literature that report, on perhaps some very interesting, but maybe rare compound present in a crop, but the impression was that stakeholders were really interested in those key parameters. You may remember that, for instance, OECD has developed consensus documents that define actually what are the important key parameters for a particular crop. So crops may differ from one to another when it comes to which nutrients, for instance, are, for instance, abundantly present in such crops, and which contribute to human nutrition, for instance. So when we have, for instance, cereals, they have a lot of B vitamins. When we have certain oilseed crops, they have, for instance, a lot of E vitamin in the oil present. And there are many more examples of this, you know, like potato having vitamin C, for instance, but also when it comes to anti-nutrients and toxins being present in those crops. The testing of newly expressed proteins in animals. So this is something that is also being recommended. You know, if a protein is completely new, if there's no experience of exposure to such a protein, then, for instance, EFSA guidance recommends to have it tested in a purified form in experimental animals. And we already knew from literature there are a couple of studies out there, particularly in mice, for instance. But the question was, you know, what is the landscape? What is the overall evidence there? There are some discrepancies here between, for instance, Europe and other regions of the world. In that, for instance, EFSA guidance recommends a 28-day, or not really 28-day, but a repeated dose oral toxicity study with the newly expressed protein, whereas in other constituencies you see that regulators already accept, for instance, acute toxicity study. Of course, we should not see this in isolation, because usually such studies are provided in combination with all kinds of other studies, for instance, digestibility studies, in silico studies and such. Okay, so I realize I spent a lot more time on explaining the background than I initially wanted. So when we had these questions, so we started, you know, when we had the protocols also ready, we started working with searching databases, and we ended up with 3 databases that we used for this purpose. Those are Web of Science, Scopus, and CAP Abstracts. So which belongs to the Commonwealth Agriculture Bureau. And we actually combined, you know, what we found using a program like EndNote, which is a reference manager-like program. So then you retrieve all the information on the authors. There are the abstracts in there, also the keywords and all kinds of other information. And usually also a link to the full text article. And based on that information, we started selecting records. So in some cases you can already see, based on the title, that this article won't actually discuss the topic you're interested in. We can come back to that later in more detail, why is that. But also the abstract, you know, if it's unclear from the title, for instance, also the abstract can provide you more details about how relevant the record is. And as you can imagine, you will never have 100% hit, you know, with only references that are relevant, because you have defined your search strings in a way that is, you know, as broadly as possible to capture most, or if not all, of the publications on a particular topic. You will also find a lot of false positives, if I may call that. So. Probably only a fraction of the total number of records that you find will be truly relevant for the question. Okay, so and that of course will provide you with the raw data. So then you will have the full references, the selected references available that you can start extracting. So you will take all the details from those references using what we call data extraction sheets. So we have, like Excel spreadsheets, and we have certain questions in there. Actually, for some evidence map, we have more than 100 rows that actually can be filled out for each specific publication. So we're trying to collect as many features as possible on each record. So the main findings and observations in these studies were that, for the whole food and feed studies, we found that there's actually a wide variety of jam crops and animal species. So the crops and the species in which these jam crops are being tested. And for this purpose, a systematic review appears feasible. So we have enough studies that are, say, sufficiently similar in a way that they can be combined in a systematic review. Regarding allergenicity, we noticed that there are actually two types of studies. First of all, there are those studies that actually investigate potential changes as a kind of unintended effect or an unintended change. You know, remembering a comment we received this morning of genetic modification. So really like, you know, whether or not something unintended has happened to the allergen repertoire and subsequently to the reactions in patients. Although in other cases we noticed that, you know, genetic engineers try to create what we call hypoallergenic crops. Okay. So in that case it was really the purpose of the modification to have a crop that causes less allergic reactions. For the key chemical crop constituents, we found that there's actually a great variety of GM crops and events, as we call them, being tested. So Monet-10, for instance, we mentioned it many times, is actually an event, but we can also have 43.2, which is the Roundup Ready soya, the first generation, or for instance the 87406, which is the drought-resistant maize, just to mention a few examples. And in all those cases we noticed that various categories of target analytes are covered. So you may have certain articles focusing, for instance, on, say, mineral composition, others focusing on fatty acid composition, some analyzing crops very comprehensively. For the newly expressed proteins, we found that there's a range of proteins being tested, but there's really preponderance of cry proteins in literature, mainly tested in rodents and usually following classical toxicology. So, okay. So here are a few highlights, illustrations from the, you know, summarizing the results. So this actually shows that in all these cases, actually a fraction of the initial number of records retrieved through these database searches was eventually selected for mapping. So varying between 37 and actually 341, as you can see, for allergenicity and whole foods. And actually, this is actually even more than we expected for allergenicity. We didn't expect to find so many publications on this topic, because this was really something that people thought, well, has it ever been explored, you know? But it certainly has. Okay. This is on GM crops being tested as a whole feed. And we see there that, you know, regarding crop species, there's a lot of GM corn being tested. Also soybean, for instance, and to a lesser extent rice. I think there are a lot of Chinese authors also doing rice. Yes, there. When it comes to the animal species to which these whole food feeds are administered, you see there's a lot being done in rat, but also in other species. Actually, this is actually before we started excluding, say, those studies that were only focused on performance. So that explains why there's still a lot of poultry and pig in there, for instance, and fish and cattle. Okay. But the rodents are already very well represented here, as expected. So when it comes to allergenicity, as I told you before, we do have studies that focus on changes in allergenicity as an unintended effect, but also a number of studies, 11 out of 37, that actually targeted, say, reduced allergenicity, so the hypoallergenic crops, which clearly is meant to, you know, benefit those allergic patients. So allergenicity, we had some additional crops here being tested as compared to the other ones, for instance tomato, but also for instance mustard. I think mustard is because of an Indian group working on, say, GM mustard and testing this extensively, you know, comparing allergen profiles with reactions in animals and patients. But also soybean, which is known to be inherently allergenic. Maize, which is not really known to be a major food allergen. It has pollen allergens, but it's not really known as a food allergen. Okay. Okay, so the whole food feed studies. The length of the studies, I hope I still have some time left.
Yeah, we've got a little bit of a buffer.
Okay, a little bit of buffer. Okay, thank you. Sorry for that, but thank you very much. So we noticed that particularly for, I guess, the subchronic studies, we see a preponderance of studies, you know, like 30, 60 days, around 90 days. A couple of multi-generational studies. Okay. Then we have the events being tested. In compositional studies, you see there's a preponderance of Roundup Ready soybean, the 43-2, but also the Monet 10. There are a lot of famous other ones, particularly, I guess, from one company, from St. Louis, but also we see that, for instance, there are many more studies, say, on events that have been studied only once or twice. Okay. Here we have the crops being analyzed for composition. So we have, you know, very well-known GM crop species like maize and soybean and cotton, but also rice and potato. So I think that this is also an interesting finding. I think particularly because we see that rice is now being developed in several Asian countries, for instance, including China. These are the analytes analyzed in those compositional studies. Oh, sorry for that. You see that there's a lot of attention, of course, for proximates. That's usually included besides the other parameters. So proximates are things like oil, fat, protein, ash, and such. Amino acids, fatty acids. So it seems to be fairly equally distributed. But it also means that, you know, that there's a certain focus in most of the articles considered. Okay, the newly expressed protein tested, as already stated, cry proteins are predominant, but also a couple of other ones related to, for instance, herbicide resistance. But also, you know, surprisingly, a few studies with a protein that was supposed to have a high biological value, for instance, for nutrition. Okay. When we discuss protein safety studies, we noticed that there's a lot of publications in a select group of journals, particularly toxicological journals and also a nutrition journal, as well as agricultural food chemistry. So I will skip the part on the safety assessment. I guess most of you are already familiar with this. And when we discuss the status quo and outlook, we're still working on the evidence maps. The idea is to publish this jointly with the protocols in a special issue to a scientific journal. We're currently negotiating with Food and Chemical Toxicology of Elsevier, and we hope to have confirmation very soon, which means we can start the review process, then submit manuscripts, assign guest editors, and hopefully be successful, because this will go through another round of peer review, but then not by stakeholders, but by peer referees invited by the journal. Another observation was that stakeholder feedback proved to be valuable. So as you may have noticed, we changed a few things in the course of the process. However, it also required additional time and resources. So, as already stated, there's the intention to have a systematic review on whole food safety based on the evidence map on whole foods. So, with my apologies for our exceedance of the allotted time, I'd like to thank you for your attention.